My name is Dr. Elizabeth Krakow. I'm a bone marrow transplant physician who works at Fred Hutchinson Cancer Research Center and Seattle Cancer Care Alliance. And today I'm joined by my colleague.
Hi, I'm Jerry Radich. I'm a clinical researcher and physician at the Fred Hutchinson Cancer Center. Basically, my lab studies what we call the genetics of luck, trying to understand who responds to therapy and who doesn't. So when a patient is diagnosed, they can have many different clones of cells. Some of those probably compete against each other. Some of those probably cooperate together. And so what we think is going on is that clonal diversity is really what drives this pattern of relapse. And I figure here that Amy Pogorgan in our lab drew, she's a much better artist than I am. And, and so it's quite beautiful. So on the left, you have patients who their disease initially responds, but what you're seeing is that one clone is going away and you get a rise in these other clones. So that's really survival of the fittest. That's really Darwinian selection. The sensitive cells are going away. Now the resistant cells have more room to grow and more food to eat. And so they come back. And then in the middle column is what Amy refers to as culling of the herd. That is, your disease populations, all the clones go down, but you just haven't treated people long enough for heart, because when you release the therapy, all of those clones come back in the same proportions, right? And then a very different mechanism is what we think may be going on with late relapses, where you initially get a very good response, but you have some, early stem cell that has some injury to it. And what you've done by giving them agents that injure DNA is you've actually caused more genetic instability, more mutations, and then these people come out later because it takes a long time for those cells to expand to 10 to the 12 cells or whatever, and they come out with relatively a different type of genotype. So I think that we have to start thinking about this in ways in addressing relapse, how to prevent it, and how to cure people, because right now, for a lot of these cases, the tumor is probably using natural selection for their advantage and against us, right? And relapse is probably completely predictable and understandable.
Now, we're in a very unique field, hematologic malignancies, because we have access to the tumor and we can sample the blood, sample the bone marrow, and detect clones when they're detectable by molecular methods, but not evident under the light microscope, not evident in the circulating blood counts. How do you think these models, starting with the easier situation of hematology, can inform our clinical trial designs?
The kind of poster child of this is called chronic myeloid leukemia, where it's actually unique in the translocation for every patient with CML will have this translocation between BCR and A disease. It doesn't need a lot of other mutations. In that disease, we have a targeted agent that actually inhibits BCR-ABL, and we can use the actual mRNA of BCR-ABL as a measure of disease burden that we can look at molecularly. So that's the kind of the perfect example where the drug target and the monitoring target's the same. And in that case, what we've done is we have now all sorts of clinical trials where what we monitor is the BCR-ABL level. So we don't need to go and look for long-term survival and the like. The actual monitoring of disease burden correlates exquisitely with those outcomes. So those have reduced in pharma trials 5-10-year randomized trials to 1-year trials, right? So now what we're trying to do is find those in other diseases. Now, in the acute myeloid leukemia space, you might be finding markers in 25-50% of patients. Only a subset of those will have drugs attached to them. And for each of those markers, that may only be 5 or 10% of the total AML population. So it's going to be very, very difficult to have kind of the classic randomized trial of standard therapy versus marker-driven therapy for a particular mutation. And so I think what we're going to have to do is almost do a change in our trial design, a principle. And that principle would be standard therapy versus targeted therapy, where anything you have a target for is put in that arm.
And that, a problem with that kind of trial design.
So do I.
For a few reasons, and we keep seeing increasing numbers of trials designed like that, right? Standard of care, some standard against a plethora of other treatments on the. There's so much heterogeneity among those treatments and among how well matched those treatments are to the targets, as you say. Sometimes we're just targeting an epiphenomenon. Yep. Sometimes, we really have a sort of, round hole, square peg situation, trying to match the target. And the drugs that we use, we can't use at the ideal doses for the target because of off-target effects as well. A lot of these targeted molecules are more promiscuous than we think, and so we're actually changing a lot about the cell signaling. And then you have perhaps 50 or 60 different treatments in your targeted arm, quote unquote. And maybe among those treatments, there are some that are very effective, but you won't see that effect if you only have a handful of people who have that target.
That may work is, the NCI is now developing this MyeloMatch precision medicine program where rapid diagnostics will be done on patients, where you can do mutation analysis within 2 days, which is kind of a new technology. And then put, as you're suggesting, specific trials up with that marker. And what's hoped is that, given using all of the intergroups, ECOG, the Alliance, SWOG, there will be enough N possible for patients to enroll in those, right? So to do that, you have to have a little bit of a change from academic centers doing their own trials to, you're asking investigators to basically do that from the goodness of the heart because they'll get very little credit academically. So that's a major shift we have to think about.
Another structural barrier to this is that in a lot of these trials, about 2/3 of patients or fewer, some of them even 1/3 of patients actually receive the treatment that's recommended to them based on the genotyping. And I think the barriers there also include insurance companies who are often unwilling to pay for a treatment that doesn't carry the FDA label for their particular malignancy.
You also have the issue that in some patients, you will have 2 markers. And often those targets will be with drugs owned by different pharma companies. And while in the HIV world, the people have gotten together and worked like that, in the oncology space, there's not a lot of examples of pharma companies taking their drug and be willing to pair it with another drug and those go into a trial because they realize with toxicities and stuff of with that combination, they may go down with.
Clinically, there's certainly situations where we've tried to do that, but there are cumulative toxicities of the order. And so how do you know which, if you have to then pair back, how do you know which one to stop? Do you have to have some understanding of which is the more causal, the more foundational mutation in your clonal model, which one preceded which in timing? Should you target the mutation that occurred earlier? Should you target the mutation that causes more proliferation? How do you know which one to pull off?
Often we kind of make assumptions by the bulk sequencing on which ones came first, and we're probably reasonable about that, but those are assumptions, right? So you have to be prepared to be burned occasionally clinically.
You talked about centralizing big teams to investigate these. Can you imagine a decentralized model where every physician treating a patient would not be afraid about protecting their medical license, but would have the freedom to experiment with that patient based on the genomic results, and in my world also based on the immune profiling after transplant, and test things, but have a system of linked electronic medical records linked to these genomic results, where if you had 100 million people in your sample.
A huge series of N of 1 trials, essentially, could be hugely informative, right? The system, the difficulty would be how to build that, right? I think from a science standpoint and from an IRB standpoint and stuff, you could build in ways to make this safe. But I think that's something that people should, I think that's something that you should do.
Jerry, thank you so much for this conversation. I
Oh,
hope
my
that
pleasure.
will give our colleagues and our audience a lot to talk about, and I look forward to their discussion.
Yep, and anyone wants to spin me an email, I'd be always happy to chat.
Same here. Thank you.
Being Wrong About Randomization by Stephen Sen. The point about designing experiments and estimation is you have to pick up the problem from the right end. The wrong end is to assume that the goal is perfect estimation. Then pretty soon you'll come across the problem of confounders. And you'll say, well, it's pretty difficult to balance everything I need to balance. And if I don't balance everything, then in that case, my estimation won't be perfect. Randomization certainly can't guarantee that all things are balanced, and so randomization is a bad idea. So if you randomize, your estimate will be imperfect, therefore randomization is not helpful and may even be harmful. The great statistician R.A. Fisher recognized the problem straight away. He understood that it was impossible to balance all prognostic factors, and that therefore perfect estimation is impossible. So we must accept that imperfect estimation is inevitable. How can we rescue this situation? Well, one way is by saying exactly how good our estimators are, and this requires that we also provide estimates of error, or perhaps confidence intervals or something similar. Randomization enables us to do this. By the way, here we have three giants of the Rothamsted School, Fisher on the top left, and then Frank Yates on the top right, and John Nelder below. Fisher also realized that it was the joint effect of prognostic factors that matter. From that point of view, there really is only one factor. The variation that this causes in outcome is what matters, and this can be measured by looking at how results vary within group. This can be used to estimate the variation between groups, which is a measure of the reliability of our imperfect estimator. This requires, however, that the variation between and within groups may be expected to be the same, and this is exactly what randomization seeks to guarantee. Here are two parables to explain the problem. The parable of the engineer and the statistician. On the left we have the engineer. The engineer builds a bridge, but the physicist, who thinks he's cleverer, tells the engineer his bridge will fail. Why? Because when the temperature increases, thermal expansion will break the supports or the bridge or both. The engineer replies, however, that he's thought about this, and that's why we use expansion joints in building a bridge. The physicist doesn't understand and says, but aren't you overlooking the thermal expansion? Very frustrating. Statistician has a similar conversation with his critics. Let's say, for example, a philosopher. It doesn't have to be a philosopher. The philosopher tells the statistician that because there are indefinitely many confounders, the estimate will be unreliable. The statistician replies that the estimate is known to be not perfectly reliable, but the confidence interval estimates how reliable it is. The philosopher says, yes, okay, but how about you dealing with all the confounders? I'm going to use a crossover trial as an illustration. It doesn't matter which one. I've got a crossover trial here, and I'm going to do two analyses. On the left-hand side, I don't fit the patient effect. I just treat it as if it was a completely randomized design. That's a wrong analysis. On the right-hand side, I do fit the patient effect. That's the right analysis. If you look at the two estimates, they're exactly the same. The difference between them is not in the estimate. The difference is in the standard error. One of the estimated standard errors is incorrect. That's the one on the left. And the one that is correct is the one on the right. The one on the right is smaller. Why is that? That's because we balance for all sorts of factors. The analysis on the left assumes we haven't balanced for all these factors, and it makes an allowance for their being unbalanced. This allowance inflates the standard error. The previous slide showed a parametric analysis, but I can illustrate the same thing using a permutation test. Here I've taken the same data and I've permutated them in two different ways. In one way, I've assumed it was completely randomized, which is wrong, and in the other, I've used the fact that it's matched by patient, so I've permuted by patient. I get the distribution of the two statistics as a result of this. One of the curves, the red curve, is the curve allowing for the fact that it's a crossover trial, and the black curve assumes that it isn't. You will see that the point estimate, which is illustrated by the blue diamond, is exactly the same in both cases. However, in the case where I have actually conditioned on the patient, it's much more impressive. It's a much further part, relatively, of the relevant distribution, and that exactly illustrates the point. The point is that the parametric analysis reflects the randomization that has happened, provided that you analyze appropriately. The indefinitely many confounders argument is a red herring. It's not the number of factors that matters, it's the sum that they make. And so people that make this particular argument are making the same error that somebody makes in saying that the series 1, a half, a quarter, etc., is infinite. There are infinitely many terms. I can let n go to infinity, and therefore the sum is not bounded. The sum is, however, bounded because the successive terms are less and less important. And this has to be the case with covariates. We cannot have an indefinite number of infinitely influential covariates. The variance will blow up beyond what is possible. The design and analysis of clinical trials does not require a perfect balance of all possible prognostic factors. It allows for their random imbalance by having randomized and by estimating their influence. This is reflected in the statement of uncertainty associated with the estimate. If we knew all prognostic factors were imbalanced, this statement would be wrong. Thus, the standard analysis of clinical trials requires for its validity an assumption that all prognostic factors are not perfectly balanced.
Yes, hello. This is Mark van der Laan. This is the second, little longer video for the roundtable discussion at the SMAC 2021 conference of workshop, really, titled Philosophy, Statistics and Health. And the point I want to raise in this as a possible roundtable discussion topic is a statistical point, and that is the comparison of what I would call SIF-based maximum likelihood or minimum loss estimators and the more general large variation of machine learning algorithms. I'm arguing in this point, at least take a stand on that to discuss, that the SIF-based approach is superior. So let's first be careful of, let's be precise how we define that. So we are talking here about, do we want to estimate from the data some functional, maybe in density, maybe in conditional density, maybe an outcome regression, maybe an optimal treatment rule, and so on. You can then define a risk function, right? Like an empirical mean of a squared error. Residual would be an example of an empirical risk, which could be used to estimate the conditional mean of an outcome given covariates. So an MLE, or minimum loss estimator, would then say, I'm going to propose a SIF, a sequence of subspaces of our assumed total parameter space. So we're typically working in highly non-parametric models for these functions we are trying to learn. And then we are saying, let's maximize or minimize, in this case, the empirical risk, minimize, let's say, the sum squared residuals over all functions in that subspace of functions. And then the subspace can grow to higher and higher complexity. And you choose then the level of complexity of the subspace with cross-validation, for example. So that's one approach. And then the other approach is much more, has a large variety of clever ideas, which you can find throughout the machine learning literature, using forward selection, backward selection, averaging, all kinds of ways of being smart and trying to do a good job in prediction. What's the big fundamental difference between these two approaches? In my view, the fundamental difference is that the MLE, the SIF-based MLE, solves score equations. Scores with respect to the loss, but let's say if it would be in density, a log likelihood loss, then it would be the actual real scores as we're used to. And an estimator of a function which solves score equations has a lot of benefits. It's the score equation which make it approximately behave like a normally distributed type thing. In particular, there are results in the literature which clearly show that if you have a SIF and you undersmooth that SIF, then under some conditions on that SIF, you can actually use that estimator to construct efficient estimators of features of the function, smooth real valued features of the function. Like you estimate a density and you care about a particular feature, which might be a G computation formula for a causal estimate. And you can plug it in after doing some undersmoothing and obtain efficient estimators. And why does that happen? Because it solves score equations. And as we know, for example, from the literature of TMLE, it's the key, the whole idea of TMLE is to make sure that your initial estimator gets updated to solve the right score for that particular feature. And that creates then an asymptotically normal or efficient estimator. Now, if the function estimator is already solving a lot of scores, then that's essentially already happening. The linear span of all these scores spans a lot of scores. So by then, with the appropriate SIF, you can actually make it efficient for lots of features. These properties you wouldn't find in the typical, more general, maybe more ad hoc type machine learning algorithms, which don't really optimize over a function space and therefore don't have that property that they solve all these scores. And as a result, you wouldn't find, all kinds of results like asymptotic efficiency of plugin estimators of random forest and so on. That's all very hard. So that's a fundamental benefit. And on top of that, it goes actually beyond that. Number one, if such a SIF MLE solves all those scores, maybe there's actually hope that for the function itself, it might have a sampling distribution which approximates some Gaussian process or type of, at least that type of inference is possible. So it's not just for the smooth features, but also for the function itself. There's hope that you can pull that off. Again, you wouldn't find that with the more general machine learning type algorithms. And then if you start thinking about how good is a plugin estimator based on a SIF-based MLE for smooth features, again, like causal effects, how good is it? Then you have, number one, you want to solve the efficient impulse curve, makes it efficient, but there's also the exact remainder, which will be nice if that's smaller. And you can show that if your plugin estimator, the one you plug in, solves a lot of scores, it starts essentially knocking out the size of the exact remainder. Like I have recent work on high order TMLE precisely showing that. So by solving a lot of scores, it's not just that it will be potentially asymptotically efficient, it will also have much better exact finite remainder with all its implications. Again, you wouldn't find that with the more general type of machine learning. Having said that, it does matter how you choose your SIF, because the typical SIFs ask for a lot of smoothness to pull it off. But for example, we now have highly adaptive lasso, which chooses as SIF all functions which are right continuous left-hand limits and have a finite variation norm. And then the variation norm is the level of complexity. And given that level of complexity, we find the best function. And that creates then the highly adaptive lasso estimator. You can use it for plugin, and we have shown it can be efficient for smooth features. But the reason why it works is because of the SIF having a special property, namely that its basis functions, the linear span of them, is a so-called Donsker class, and therefore remains a very controlled class. Most algorithms get into overfitting. So they don't really optimize for a given level of complexity, and that pay a price for that. And as a result, you cannot pull off these kind of results. For highly adaptive lasso, we have a rate of convergence for the function itself of n to the minus 1/3, under essentially no conditions, which is remarkable. You wouldn't find that very easily, at least not that I'm aware of, for many other SIF-based max select estimator, except if we go to a very high level of smoothness. And n to the minus 1/3 is a dimension-free rate. So it does matter how we choose the SIF. And so all of this raises then also the question, the Bayesians. The Bayesians also work with SIFs and do then essentially max likelihood, except they do it with the posterior distribution. So for them, it matters too. So I think they're a very interesting question. And I also think it goes back to really having to understand the theory in order to really figure out what the right approach is. Because it does make a real difference, and it's not obvious always a priori to know why. And so anyway, I want to just share some of the things I've learned over the last years, which certainly have implications for my thinking about super learning, what to include, and so on. Anyway, I'll leave it at that. Thanks so much.
Hello. I have to discuss with you about the question, what do statistical tests really say? And this in 6 minutes. And it is impossible, and this is good news. First, statistical tests at the beginning, it is like a Shakespeare piece. With characters, the father, Carl Pearson, the son, Egon Pearson, the genius, Fisher, the mathematician, Neyman. And all these guys are working in the same place, and they all love each other. Of course, this is completely wrong. There were tension because of power, etc., etc. Timing at the beginning of the 20th century up to 2021 in Toulouse, because it seems that the question is still open. Audience, millions, tens of millions of students, teachers that try to learn how to do and interpret statistical tests. So, if we look at what proposed Fisher in 1922, first he considered a hypothetical infinite population, and this is crucial. There is somewhere a hypothetical infinite population. Then what you have is a sample, it is a data set. And from this data set, you estimate the probability that a worse fit should be obtained from a random sample of the hypothetical infinite population. And this is the p-value. If the p-value is equal to 1%, then it is very unlikely that the sample comes from a random sample of the hypothetical infinite population, and so you can say no with a certain risk, of course, or with a certain improbability, which is estimated by p. For Neyman and Pearson, in fact, in their paper, 6 years after, their proposition seems to be quite different. They are looking for rules to govern behavior. And you know that with Neyman and Pearson, indeed, there are H0 and HA. The notation were different at that time. And you will have to accept one or the other. And doing that, you are likely to take two risks, type 1 and type 2. And their objective is to fix type 1 error and to minimize type 2 error. So apparently it seems very different from Fisher. But in fact it is a little bit close, because in both situations you estimate a p-value, and with Neyman and Pearson you compare the p-value to alpha. So for many people, after the beginning of the 20th century, they say, oh, this dispute between Neyman and Pearson and Fisher, this is only a matter of ego and not really something serious. I don't think so. I don't think so, and this on very practical grounds. Why? Because, you know, when we teach statistics, there is always the question of, is it possible to accept H0? For Fisher, he writes that, he is very clear, this is not possible to accept H0. While for Neyman and Pearson, indeed, if you have estimated the type 2 error, you can accept H0. The risk is equal to beta. Another point that is quite tricky when you teach is the question of one-sided versus two-sided test. And why it is tricky? Because for Fisher, you have seen that the test is basically rely on a goodness of fit test, and goodness of fit tests are mainly relying on the two-sided formulation. While for Neyman and Pearson, you have H0 and HA, and according to the sign of HA, you will have one direction or the other direction in the decision, and so basically Neyman and Pearson tests are one-sided, while Fisher tests are two-sided. Third point, you know, and this is it often happens in the real life of experiments. If you have a p equal to 0.048 and p equal to 0.052, should the conclusion be really different? And most people will say, no, it is very close. And indeed, for Fisher, it is the case. While for Neyman and Pearson, no, because it is a behavior. And according to the position in comparison to 0.05, you will accept or reject H0. And so the two values will lead to very different conclusions. And indeed, Fisher and Neyman and Pearson acknowledge their difference, saying that Fisher was dealing with inductive inference, while Neyman and Pearson with inductive behavior. Okay, but what is induction? Philosophers do not totally agree with that. Basically, induction, it comes from the particular to the general, and in fact, it is a little bit true, especially in the way Fisher describes his test. From the sample, you go to a hypothetical infinite population, from the particular to the general being a hypothetical infinite population. But what is this hypothetical infinite population? It is very speculative, and this is the reason why you could say that statistical induction is not more than a fantasy, a useful fantasy, but a fantasy. And in particular, what don't say statistical tests, the truth, of course, while many people think that, and many people say that because we are guilty statisticians. You know, we encourage people to misuse statistical tests. Just look at statistical tests of normality. If you are, according to Neumann person, because you want to accept H0, you should need to estimate a type 2 error that nobody do in reality. So people are using Fisher inference, but with Fisher inference, you cannot accept H0. And this is H0 that you want to show when you are testing normality. So testing normality shouldn't be done in practice. So to conclude, how should we use statistical tests in medicine in particular, which is my job? What does they say in medicine? If you are doing public health, indeed, you can use the Neumann person perspective. And this is what does the FDA, the Food and Drug Administration, and the European Drug Agency. In the research, more general research setting, you can see what said Fisher. This is very enlightening. This is very emotional. He says that we have the duty of formulating, of summarizing, and of communicating our conclusion in intelligibly form, in recognition of the right of other free minds to utilize them in making their own decision. And so for Fisher, you do test to communicate conclusion so that other people will be able to take their own decision. And this is what we should say when we teach statistical tests to students. Now for clinicians, this is likely to be the most tricky situation, you know, because you have in front of you a unique person and you have inference for what? In average. So this is quite impossible to say to a clinician how to deal with statistics with a unique person. The only way I have found is that if you live in a linear world, then the average effect of a treatment is equal to the effect on an average people. And so, in a linear world, clinicians are living like in the Minecraft video game, where human beings are with big pixels, big voxels. And these voxels correspond to the variable on which you condition the estimate of the efficacy of the drug. And so, physicians should be aware that when they are doing evidence-based medicine, which is absolutely crucial, they do not have anymore a real human being in front of them, but some kind of a body in the Minecraft video game. Thank you for your attention.
A main source of today's crisis in replication is that high-powered methods make it too easy to uncover impressive-looking findings, even if they're false. If little or nothing has been done to rule out flaws in inferring a claim, we don't have evidence for it. It hasn't passed any kind of a severe test. So the first question to ask in scrutinizing a statistical account is, does it permit violations of this minimal requirement for evidence? A second question concerns presuppositions about the role of probability. In frequentist error statistics, probability is intended to assess and control the probabilities of misleading interpretations of data. Now, to those who assume probability has to provide degrees of belief or probability in hypothesis, a p-value appears to be irrelevant or necessarily misconstrued. But that's to presuppose a rival philosophy of inference, akin to Carnap insisting Popper's falsification has become a confirmationist. So the second question to ask is, does an account enable falsifying claims, at least statistically? We often hear it's just too easy to obtain small p-values, and yet replication attempts find it difficult to get small p-values when they have pre-registered hypotheses. What this shows is that the problem isn't p-values, but failing to adjust them for cherry picking, multiple testing, and other biasing selection effects. A third question is the uncertainty measure directly altered by biasing selection effects as we think they should be? The same data-dredged hypothesis can occur in methods put forward as alternatives to p-values, likelihood ratios, Bayes factors, and others, except now we lose control of the grounds to criticize inferences for flouting error control. Finally, even those who criticize p-values will employ them, at least if they care to test the assumptions of their statistical models, an altogether important task. So a fourth question is the account capable of testing its own assumptions? So if someone is selling you an account or a reform where the answer to any of these questions is no, you may wish to hold off buying it. Thank you. I think that many of the reforms in order to improve on replication are good. For example, you shouldn't be using statistical tests in an unthinking cookbook manner that has long been derided. They should be thoughtfully used and blended with empirical and theoretical background ideas. Then there's also the idea that we ought to pre-register our results, our hypothesis, so that somebody can critically evaluate the probability that you would have found something that looks impressive, even if it's due to chance. So there have been some good reforms, but others are quite radical, and I say even can obstruct practices known to improve on replication. And that's why it's so important to understand the philosophical basis for some of these arguments. I can give you an example. The American Statistical Association put out this statement on p-values in 2016, which really did just rehearse a lot of the claims about what a p-value is and how to avoid p-values. Not a posterior statistical significance, is not substantive significance. No evidence against a claim is not evidence for it. Well-known claims, but then a few years later in 2019, some of the same authors put out an editorial which was sort of presented as if it was just a continuation of the 2016 statement, but it was quite radical. It actually said it's time to stop saying statistical significance altogether, and it's time to not use statistical thresholds in interpreting data at all. And I actually think that these suggestions, although I understand where they may be coming from, defeat some of the most important things. Because if you don't have a threshold, okay, then you really don't have a test. It's necessary that at least some of the outcomes won't be allowed to count as evidence in favor of a claim in order to have a test. And that's tantamount to having a threshold. I mean, so this is this big debate, and I don't know whether you want to get further into it or not, but basically, I think some of the reforms that are thought to be good things, after all, you might say, if you don't have to meet 0.05 or 0.001 or whatever threshold is, then maybe you're not going to be lured by these so-called perverse incentives to try to cherry pick and data dredge. And that's, of course, the key problem behind non-replication. But in fact, a researcher is still going to want to show that they have evidence of a real effect. They're still going to have to, in effect, show a small p-value. To take a non-statistically significant p-value and claim this is good evidence for an effect would actually not make sense. It would be to say that even though the probability of larger and more extreme differences than I got often occurs by chance alone, I claim this data are good evidence for non-chance. So what happens is that the researcher, still wanting to show an impressive effect, would be led to data dredge, to change the endpoint, and so on. The only trouble is, without the thresholds in place, it would be very hard to hold them accountable. So I say that it would actually obstruct practices known to improve on replication instead of helping things.
Hello, my name is Antoine Chambaz. I would like to thank the organizers of the SMAC 2021 workshop for giving me the opportunity to participate to the Crazy Friday Roundtable that will take place on June 18th. It truly is an honor to sit at this virtual table with Elisabeth Krakow, Deborah Mayo, Bruno Falissard, Sander Greenland, Michael Kosorok, Siben Sen, and Mark Vandelan. I wish we really were in the same room. My argument unfolds in 5 successive steps. In step 1, I will describe briefly a universal template for statistical analysis. In step 2 and 3, I will share with you a personal experience. In step 4, I will discuss about deterministic models. In step 5, I will discuss about assisting medical practice, and then I will be in a position to ask you my questions. One of the universal templates to conduct a statistical analysis unfolds like this. First, a set of questions are formulated on a phenomenon of interest. An experiment more or less closely related to the phenomenon is either found or designed, and an observation drawn from that experiment is collected. Second, the statistician defines a model M for the law P of the experiment, justifying its construction based on knowledge available on the phenomenon. Then she introduces a statistical mapping psi tailored to formalize the questions. Third, she analyzes psi, the analysis revealing what are the features of P that really matter for the estimation of the parameter of interest. Let's call Q those features, where the parameter of interest is the value taken by psi at P. She constructs an estimator Q hat of O of Q, typically using machine learning techniques. She then constructs an estimator psi hat of O targeted to psi of P, using Q hat of O and preferably under the form psi of pi, where pi belongs to the statistical model M. At this stage, she carries out the theoretical analysis of psi hat of O and a numerical analysis as well, based on simulations. If applicable, she derives confidence regions and R tests. Finally, the statistician shares her results and the conditions under which they were obtained, and she discusses them with the interested scientific communities. I wish to focus on the definition of the model and on the estimation of the features of interest, Q, by playing on a Gallicism-based pun. The French word for experiment is expérience, like the English word experience. Now allow me to share 2 personal experiments, expérience, with you. The first experiment, expérience, starts with this painting by French artist Jean-Pierre Schneider. I am particularly fond of it. I find it beautiful beyond measures. It moves me deeply, and in fact, it resonates powerfully with me, in particular on a professional note. The numbers, the cross, the red balls, the whiter manifolds, the contours slightly suggested, and the black background makes me think of a statistical law. My love for this painting also follows from its place in history, and the tribute that the artist pays to Manet's celebrated Olympia, and to the black character and model who stands by the bed and brings flower to the other woman. In contrast to Manet's painting, Schneider's painting also seems livelier in the sense that it leaves room for many more stories to be told. The second experiment, expérience, that I would like to share with you revolves around another celebrated painting, Danseuse montant un escalier, Dancers Going Up a Flight of Stairs, by Degas. When I first read the painting, if I may say so, I naturally decomposed it in a series of synchronous snapshots arranged horizontally. To my eyes, the still image, bathed in soft light, captured gracefully an instant in time. Later, I learned by chance that perhaps one single model posed for Degas, and then my reading of the painting changed dramatically. I now naturally decompose it in a series of successive snapshots arranged vertically. The image was not still anymore but animated, the soft light flowing from bottom up and left to right, like water. Now that I've shared these personal experience with you, let's go back to more mathematical ground and discuss the notion of deterministic models. Countless deterministic models have been developed to model natural phenomena. In this brief talk, deterministic models should not be confused with statistical models. What I call deterministic model is a mathematical model obtained by combining elementary blocks, each block being based on the fundamental laws of physics. Such a model provides, possibly at several scales simultaneously, a mathematical description of a phenomenon under the form of equations, the equations involving quantities that can be observed or not. A collection of physico-chemical, physiological, or environmental parameters allow to characterize the regime of the model. Consider this lung, for instance. It is a complex natural system. There exist several deterministic models describing how it functions, with different levels of granularity. One of the simplest models consists of two blocks. The first block, sketched in this image, draws a comparison between a lung and a mass spring system. The second block, not shown, focuses on the exchange of gases that occur through the alveolar capillary membrane. The deterministic model is quite precise, in the sense that it allows to reproduce quite faithfully the functioning of lungs under a large range of conditions. However, it must be acknowledged that the description is, by design, stationary. Statistical methods and deterministic models can be helpful to assist medical practice. The experience of a patient is multifaceted and extraordinarily complex. Today, large amounts of data are routinely gathered, in particular in intensive care units, and many questions naturally arise. Numerous statistical methods have been developed and studied to assist the medical teams at the bedside of patients, and in parallel, numerous deterministic models have been developed and studied to model human organs. Those deterministic models lend themselves to being combined in sophisticated hierarchies of models. And I am, at last, in a position to ask you my questions. Do you, like me, think that statisticians should learn to rely on deterministic models? And if so, how? I thank you for your attention.
Hello, this is Michael Kosrock from the University of North Carolina at Chapel Hill again. I'd like to ask an additional question. Is the field of data science ready to solve the world's problems? The field of data science is an amalgamation of many disciplines, including what I would like to call the foundational disciplines, and these are computational sciences, informatics, mathematics, and statistics. And these are crucially important in the quest to improve the world through wise use of data and information. Each of the foundational constituent disciplines brings crucial perspectives and capabilities to the table. But are we flexible and deep enough in the way we are thinking about the foundations of data science? Are we working sufficiently well with applied domain experts and key stakeholders in society to maximize benefit and minimize harm? Are we inclusive enough in how we collaborate with application domain experts? The focus of the field of statistics, for example, is inference, which generally speaking refers to the question of how conclusions from data will have meaning in the real world. This is the central focus of statistics, and it has a long tradition of models and frameworks for answering these questions. However, we are at the crossroads of a potential major transition in the field. The essence of this question at the crossroads is whether we are willing to sufficiently adapt as a field to rise to the occasion that presents itself. These questions include the following 4 examples. First, can we expand our theoretical foundations, methods, and applications to be sufficiently broad to handle all data and analytic situations which may present themselves that require inferential reasoning, or things like inferential reasoning? Are we ready to better teach and train the world on the fundamentals of inference, inferential tuition, and practical quantitative reasoning, including teaching other data scientists, domain science experts, policymakers, and society at large? 3, Are we prepared to work with new sources of data and information, including qualitative and non-stochastically generated samples, for example, and data that occurs in real time? 4, are we willing to take on collaborative leadership to a greater degree as needed? Similar questions arise in the other constituent foundational data science domains, and we all need to proactively prepare for the coming explosion in data science and its applications.